Ever wonder if you could genetically frame your identical twin?
Every human has 46 chromosomes. 23 chromosomes come from their mother, and the other 23 come from their father. Altogether, these chromosomes contain genes. But what are genes? Genes are a basic unit of DNA that control the development of one or more traits. In 1953, James Watson and Francis Crick discovered the structure of DNA with the help of Rosalind Franklin and Chargaff's research.
So what does the structure of DNA look like? DNA has a spiral ladder that is called a double helix.
This structure is made up of phosphate molecules, deoxyribose sugars that form the backbone of the ladder, and the nitrogenous bases form the rungs.
But what are nitrogenous bases? Nitrogenous bases are nucleotides. Nucleotides are the four building blocks of DNA. These four building blocks are adenine, cytosine, guanine, and thymine.
According to Chargaff's rules, adenine is paired with thymine, and guanine is paired with cytosine. These base pairs are joined by hydrogen bonds. Thymine and adenine have 2 hydrogen bonds, and guanine and cytosine have 3 hydrogen bonds. And in total, there are 3 billion bases in human DNA. DNA, which is double-stranded, can be compared to an instruction manual that cannot leave the nucleus without risk of being damaged. So a copy called RNA, which is single-stranded, is made through a process called transcription. After transcription occurs, the RNA leaves the nucleus and attaches to ribosomes in the cytoplasm. It then uses the RNA chain to add amino acids and create a protein chain. This process is called translation, and is used to build proteins in the cells, which in turn display the genes we see in all living things. Another difference between DNA and RNA is that the nitrogenous base thymine in the code is replaced with uracil. In October of 1990, plans were developed to begin the process of producing maps of the entire genetic makeup of a human being. This project was called the Human Genome Project. With the collaborative work of many scientists around the world, the gene map was completed in May of 2000, consisting of genes like hair color, eye color, height, and 30,000 other genes. In the debate of nature versus nurture, a person's genetics is their nature and their environment is their nurture. At times, it is difficult to identify how much a person is determined by their genes, nature, and how much is determined by their environment, nurture. All living things have their own genetic makeup, whereas identical twins share 100% of their genetics, meaning that they were fertilized by the same egg and the same sperm. In contrast, fraternal twins and siblings only share 50% of their genes, meaning they were fertilized by a different egg and a different sperm. So, if one twin were to frame the other and the police were to find any DNA evidence such as hair, saliva, blood, etc., it would be very difficult to determine which twin the DNA belongs to. The main distinctive difference between identical twins is their fingerprints. Identical twins have distinct fingerprints because both genetic and environmental factors contribute to fingerprint formation in the womb.
In the south of France, 6 women were raped. Evidence, including DNA, led police to not one but 2 suspects, identical twins Elwin and Johann. When asked to identify the attacker, victim recognized the twin but couldn't say which one had assaulted them. Scientists were then brought in to help distinguish which twin committed the crime. The human genome consists of a 3 billion letter code, says George Grandel, a sequencing expert. If the body is growing or an embryo is developing, then all the 3 billion letters have to be copied. During this copying process in the body, there are typos happening, says Grandel, referring to slight mutations. In standard DNA tests, only a tiny fraction of the code is analyzed, enough to differentiate between 2 average people like siblings, but not identical twins. Grandel and his team took samples from the male twins and looked at the entire 3 billion letter sequence, and they found a few dozen differences in the DNA. These genetic mutations happen because of epigenetics. Epigenetics refers to the external modification of DNA that turns certain genes on and off. This modification of DNA is caused by someone's environment. This is how twins who have identical genetic makeup end up with different mutations in their genes that distinguish them from one another. The scientist also tested the son of one of the men and found that he had inherited 5 of the mutations from his father. Therefore, because of the new advances in science that have occurred, it has become much more difficult to frame your twin now than 20 years ago when they didn't have the same technology as now. Going back to the initial question of ever wonder if you could genetically frame your identical twin, the answer is no, thanks to the evolution of science. To all you twins out there, don't think you can get away with framing your twin now.